I want to, oh, just getting the recording notice. Okay. I want to begin by acknowledging with respect the Lekwungen peoples on whose traditional territory I have the privilege both of working on and living on in my home, as well as the Songhees, Esquimalt, and Wasanich peoples whose historical relationships with the lands, and I always like to add the waters as well, continue to this day. So I'm really thrilled to be able to speak with you this morning. I wish it was a little bit more interactive. I know these Zoom seminars are challenging. And as Boris said, my roots are at Dalhousie. And so I want to also just acknowledge the amazing mentorship that I had during the many years that I was there doing my master's and PhD. I was fortunate to be supervised both by Ransom Myers and Jeff Hutchings. And their support of me was just absolutely fabulous and really set me on the path to my success in my career because of that amazing mentorship and support, as well as mentorship from Boris, who was a great mentor to me during my PhD, along with Heike Lotze. So I want to acknowledge all of those, where none of us gets to where we are without a lot of privilege and support and mentorship along the way. So I'm going to talk this morning about ocean climate change impacts, and then I'll finish with a little bit about solutions, which is an area that my lab is transitioning to now. And I also want to acknowledge the wonderful group of graduate students and postdoctoral fellows that I have the privilege of working with at the University of Victoria. This is probably a familiar-ish photo. This is how we interacted for a year and a half. During COVID, we are back more or less in person now, and we were able to do our annual lab retreat. We missed it during the first year of COVID. We were able to do it this Labor Day weekend out at Bamfield Marine Sciences Center. Can you see the arrow when I point to it? This is perhaps one of our youngest graduate students, the little one that I spawned. And at Dalhousie, my roots with Ram and Jeff, probably not surprisingly, were in fisheries. So thinking about the impacts of fishing and often overexploitation on the ocean. And so I'll just show you some of the work that we're continuing in the lab to this day that still pertains to those topics that I studied during my graduate work and subsequently with Boris. So these are some of the recent papers. We were looking at the impacts of COVID on fisheries in the US. We've continued to work on sharks a little bit. Our roots really are on fish and fisheries. So I'm going to talk a lot about corals today, but I'm really a fish person at heart. And we've been working on fish locally in the Fraser River estuary, which is where Vancouver sits, and in temperate eelgrass systems as well, and as I said, a little bit more on sharks. Beyond these areas and the main focal areas of my talk today, an area that's really near and dear to my heart is trying to enhance equity, diversity, and inclusion in STEM. And this is something that I really I guess was very privileged and fortunate to be able to sail through my entire graduate studies at Dalhousie, never really thinking about this issue. But as a woman in science, as one goes further and further in one's career, you start to hit the glass ceiling harder and harder. And so this is something that's really near and dear to my heart, and at times very infuriating problem, but one that I've tried to turn into action with some papers recently. So this was a commentary that my friend Tara Martin and I wrote a couple of years ago in response to a rather egregious article titled 100 Articles Every Ecologist Must Read, which featured only 2 out of 100 women. So we had a few things to say about that. More recently, this year, again, this was in response to an article, this time an article that said that graduate students suffer when they have female mentors. So we had a few things to say about that as well. And this was a large, really beautiful collaboration led by Sarah Davies at Boston University, as well as by Amanda Bates, who's my new colleague at UVic, where she's just come and taken up an impact chair. And so we were pointing out here the narrow view of scientific impact, which often focuses only on citations, and urging a broader, more inclusive lens on academia. And then I've just finished writing a book chapter for a forthcoming book led by Larry Crowder on navigating to solutions in marine conservation. And this is a chapter that I have the privilege of writing with a really talented graduate student at Duke University, Leo Gaskins. And so these are some of the recommendations that we make in the chapter for recruiting diverse people into marine conservation, and then making it a more hospitable place for them once they are in the field, thinking about ways to retain, empower, and champion them. Okay, so turning to climate change. This is really a figure that is blazoned in my mind and I think about a lot these days. And it's one that has become rather famous over the past year. It was developed by climate scientist Ed Hawkins. It's known as the warming stripes. Also, people have COVID masks with the warming stripes on now. And it's depicting for each year between 1850 and basically almost up to the present, the annual mean global temperature relative to sort of a baseline period between 1971 and 2000. And so if, like me, you were born in the mid 70s, you were born in sort of this cool period, we still remember very cold winters. I remember very cold winters with lots and lots and lots of snow in Ontario. And if you were born and are maybe in finishing undergraduate or in graduate school now, you were born somewhere around here, which means you actually have no normal baseline. And so we talk a lot in ecology and fisheries about the shifting baseline syndrome. And I think we have are in danger in that of that with respect to climate change as well. So as you can see, and I'm sure you're all aware, we're in this in the midst of this period of very rapid environmental change on the planet, both on land and in the ocean. And this past decade, we've had most of the warmest years on record. So climate change doesn't, however, just happen in this very slow, gradual way, this gradual warming. Climate change also is increasing the variability of climatic events. And with that, we're getting these extreme weather events, things like very intense heat waves, like we had out here with the heat dome in British Columbia this past summer. Those can often lead to wildfires. We also experience extreme droughts in the prairies in Canada, and just in the past couple of weeks, the record flooding in British Columbia. So I had just returned from COP26, the climate summit, and returned to this extreme flooding in British Columbia. So these extreme events are all around us. And I think it's these extreme events that really serve as a wake-up call to us that climate change is happening now. It's no longer a distant threat. The climate emergency is right now. And for me, that the extreme event that was the wake-up call for me about climate change and really shifted my entire research program from having focused for a couple decades on impacts of fisheries to now focusing on climate change was the 2015-2016 El Nino event. So what you're seeing here is the Pacific Ocean. El Niños tend to sweep across the Pacific from the coast of South America. And this black circle here is the location of my long-term study site, Christmas Island in the equatorial Pacific. And so I'm going to tell you about what happened there during that El Nino. So El Niños are, of course, a natural oceanographic phenomenon, but we also know that they are increasing in frequency, in intensity, and in duration with climate change. And in the past couple of years, these and other types of pulse temperature anomalies have been termed marine heat waves. There's been lots of studies of them recently showing these increases with climate change. And there's also been studies so that we can now confidently attribute them to human-induced global warming. Okay, so I'm going to take you to Christmas Island and introduce you to the island and then show you how the 2015-2016 El Nino unfolded there. So Christmas, as I said, and as I showed you a minute ago, is in the central equatorial Pacific. It's actually just above the equator. People always say, like, have fun in your beautiful South Pacific tropical island adventure. And we always kind of laugh because It's actually not in the South Pacific, and it's, I don't know, people picture that we're going to Club Med or something, and that nothing could be further from the truth. It is the world's largest atoll by land mass. So it is about 150 kilometers in perimeter, quite large. And I'll highlight for you the relevance of that in a minute. First, I want to show you just a few photos from the island. So we're, of course, marine ecologists. We're studying the marine life underwater, which is where we spend the vast majority of our time. But Christmas is also one of the Pacific's most important seabird colonies, which is a really amazing sight to behold. And this is sort of the vantage point that we're usually seeing the island from. And now the relevance of the size of the island is that most people on the island live on the northwest coast. And so this creates a very strong spatial gradient of local anthropogenic pressures. And that allows us to look at how reefs are impacted. Initially, my main interest was trying to understand how reefs are impacted by fishing. So very close to the villages, this is what the reefs look like, if you can even still term this a reef. There's maybe a couple of live corals there. And at the other end of the island, the most remote distance from the villages, the reefs looked like this. So stunning coral reefs with high coral biodiversity, very high coral cover, and lots of fish communities. And at least when I first started working there, lots of reef sharks as well. So we've been working there since 2007, running a coupled ecological and socioeconomic monitoring program. And through that socioeconomic monitoring program, we have quantified local anthropogenic pressures all around the island. What you're seeing here are 40 monitoring sites where we do monitoring of the coral reefs, all at about 10 to 12 meters depth. And so we have this really beautiful spatial gradient of disturbance. So over the course of those 15 years or so, we've done a lot of different types of studies. And until that El Nino, all of them were focusing on impacts of fishing and impacts of those local anthropogenic pressures. As well as outreach with the local school kids. Okay, so with that backdrop and setting, this El Nino was predicted to be coming in 2015. And that presented this really amazing scientific opportunity to try to understand multiple stressors. So thinking about not just climate change, but climate change interacting with local anthropogenic pressures. And the reason, of course, that I'm sure everyone knows this, but I'm just going to do a very quick little biology lesson on corals. So corals live in symbiosis with single-celled photosynthetic dinoflagellates from the family Symbiodinaceae. There's been a lot of taxonomic revision of these symbionts over the past few years, and it's still ongoing, but many of us probably learned about these symbionts that live within corals under the term Xanthellae, which they're no longer known by. These organisms live within the coral tissue and they photosynthesize. So this is a nutritional symbiosis. They're providing the coral with their main food source, and in turn, the corals are protecting the microalgae. Now, under a microscope, all of the different kinds of symbionts all look the same. And so until we had molecular techniques, people thought that there was only one type of symbiont living within corals. We now know that there are tens, if not hundreds, of different types of symbionts living within different corals. And these different types of symbionts really range almost all the way from parasites to mutualists. And a lot of that has to do with the extent to which they provision their host with energy. We also know that these symbionts have really highly variable environmental sensitivity, so different physiological tolerances to environmental stressors, and in particular heat stress. And so this, the coral symbiosis is really at the heart of a lot of research right now pertaining to the impacts of climate change on corals, because it's believed that it might hold the key to coral resilience under climate change. So heat stress disrupts the intimate symbiosis that fuels corals. And that's the process known widely as coral bleaching. And so if you start with a healthy coral and then some sort of environmental stress happens, here we're thinking about heat stress, you get the breakdown of symbiosis. And as that symbiosis breaks down, the corals eject the symbionts from them, and then they're basically on the path to starvation. If the environmental stress, the heat stress abates quickly enough, often within a couple of weeks, the corals are able to reestablish the symbiosis and they can recover from bleaching. But if this stress persists for long enough, the corals will starve and eventually die. And again, this is really highly variable amongst different coral species, but not just amongst coral species, which is one of the mysteries and things that a lot of research is trying to sort out at the moment. But we also see a lot of variability even within the same species, and even when the same species is subjected to the exact same environmental conditions as you're seeing in this photo here, where we've got a branching Posilopora coral that's bleached and one right beside it that is not. So my idea for the 2015-2016 El Nino was that in order to understand which corals are resilient to climate change, they would be subjected to this massive stress test. So this natural heat stress. Most of this research in the coral world is happening in laboratories where corals are being subjected to very short duration heat stress assays. So I'm much more a field ecologist than I am a lab ecologist. And so I was really thinking about this as a study design of a natural heat stress experiment. And the idea was that we would go out, tag hundreds and hundreds of corals all around the island, follow them through this heat stress event, take tissue samples of them at every time point and run molecular sequencing analysis on them to determine what type of symbionts were in them. And the idea being that hopefully we would be able to pinpoint why some of them survived the event and some of them perished. So this work took place with a whole bunch of amazing students and collaborators, and I'm just going to highlight a couple of the students: Danielle Clark, Christina Tijan, and Jen Magel, who were absolutely fundamental to pulling off what ended up being 7 different expeditions to the island over the course of the heat wave. From before it, through it, and slightly after it. And incredible collaborators, including Kim Cobb, Ruth Gates, Melissa Guerin, Andrew Baker, Ross Cunning, and John Burns. And so I'm going to show you what unfolded on Christmas during this event. So this is this global map. Christmas is right in the middle there. So you've got the Hawaiian Islands up there in the Northern Pacific, and Christmas is just due south of Honolulu. And what I'm going to show you is the progression of the heat wave from January 2015 through till the spring of 2016. And what you're going to see is that the heat stress lands on Christmas Island by about May 2015. And the heat stress from that El Nino happened, was experienced in many other parts of the world's oceans, but in most other places, it landed on them, persisted for a while, and then moved off. Whereas on Christmas Island, it just landed on it and got a chokehold on the island for 10 straight months. So just for reference, corals can bleach after only 4 weeks of heat stress. Coral mortality is generally expected after about 8 weeks of heat stress. And here the heat stress lasted for 10 months. So we weren't even talking about weeks anymore. We were suddenly into this brand new territory of months. This duration of heat stress had been predicted that it would not occur on any coral reef until mid-century. And so we were in this, completely unpredicted scenario with heat stress of a duration that had never before been seen on any coral reef on the planet. Okay, so just to show you how luxurious our whole setting is when we're out working on Christmas Island. This is our beautiful research vessel that we work from. This is the back end of it. So we're packed in there kind of like sardines with all our dive tanks. This is the front end of it, and you can take a guess as to who are the rookies on the team and who are the experienced scientists experienced at working in the equatorial sun. We're diving, we're all day, we're processing samples on the boat in between dives. This again is as underwater. And then in the evening, we're processing samples pretty late into the night, every night. Okay, so this is what we saw, and I'm going to show you the progression of the event here. So July 2015, we were about 2 months into the heat stress of the El Nino. And at that point, there was variable bleaching. So some coral species had started to bleach. Again, you can see this variability within species. So these are acroporas. This one is bleached and this one is just barely starting to bleach. But a lot of species hadn't even started to bleach, or there was very little bleaching. By Christmas that year, December 2015, 7 months into the heat stress event, there had been extensive bleaching and a lot of the corals had died. So you see the bleached corals in the foreground there, but what you see behind you are corals that have already died. So they've bleached, they've died, and then they've been overgrown by very short filamentous turf algae. And at the timing of our next expedition in March 2016, so this was end of March, beginning of April, so right towards the tail end of the heat stress event, 10 months in, this is what we found. So reefs that were almost entirely dead. This is one of the large acroporid corals, known colloquially as the tabletop coral, because they are absolutely massive. And we know, so the previous largest El Nino was the 1997-1998 El Nino. It also struck Christmas Island hard, but we know that all of these corals survived that major El Nino because these corals are at least 60 or 70 years old. All of them perished without exception in this event. And we had photo quadrats, so we take photos, have been taking photos at all of our monitoring sites since 2007. We analyzed those back in the lab, identifying hundreds of points from each site, from each expedition. So this very simple, boring looking box plot is actually a reflection of over 300,000 different points of the benthic community composition on the reef, spanning between 2007 and 2016. And what you can see is the enormous loss of hard coral cover during the event. What you can also see is the large amount of variability beforehand, and that's because of that local disturbance gradient. So across the reef beforehand, there were very significant differences in the amount of coral cover at sites that were exposed to very low levels of anthropogenic disturbance. So those very high coral cover reefs that I showed you at the beginning of the presentation, versus reefs that were right near the villages and exposed to higher levels of anthropogenic disturbance. But consistently across the entire reef, almost all of the coral cover was lost during these 10 months of heat stress. So this, I'm just going to show you the transformation at one of the sites. This is one of our sites that had been exposed to very little human disturbance prior to the event. And you can just keep your eye on this very distinctive looking acroporid coral. And this is the transformation that it underwent. So overall, we estimate that almost 90% of all of the hard corals were lost during this event. So often when people are talking about the impacts of heat waves on coral reefs, they're talking about the percent of corals that bleached. There was a lot of bleaching, for example, during this El Nino on the Great Barrier Reef, and that was really widely reported. But the heat stress that was experienced on Christmas was several times more than that experienced on the Great Barrier Reef. And so here we're not just talking about bleaching, we're talking about the complete mortality of most of the reef. Now, of course, not all corals were impacted equally. Some of them had much higher survival than others. And so what that leads to is these very huge changes in coral community composition. So under climate change, we are losing enormous amounts of coral, but we are also transforming what species of corals persist on the reef. And of course, different species provide different ecological structure, for example, and complexity, and so different ecological services, really. And so what I want to point out here is that prior to the heat stress, there were different coral community compositions at the two ends of this spatial disturbance gradient. So at the very low disturbance end, the community was really dominated by corals that are regarded as having competitive life history strategy. And at the very high disturbance end of the island, the community was dominated by corals regarded as having a stress tolerant life history strategy. So probably not surprisingly, what happened during the heat stress event is that more of those competitive corals were lost. They're highly susceptible to heat stress, and the stress tolerant ones persisted. And so we really have coral communities now that are dominated by few species. So I want to just highlight a couple of my other students' work. So of course, coral reefs are not only comprised of hard corals, there are also soft corals on the reef. And one of my recent master's students, Dominique Malchieri, really took a shine to soft corals. I love to see students get really excited about certain things. We have, of course, these massive data sets that we've been collecting over 15 years on Christmas Island. And Dom was one of those students who had to do her entire thesis during COVID. She was identifying corals in all of the photos, and really felt like the soft corals were the underdogs. No one was focusing on them. She did a literature review and found that of all of the papers documenting this 2015-2016 El Nino, less than 5% actually focused on soft corals. So she focused on the soft corals on Christmas Island, and what she documented was the complete obliteration of them. So absolutely no soft corals were left, at least in our particular study sites after the event. And we didn't detect any new soft corals at our sites until 2019, 3 years after the event. So soft corals are highly vulnerable to heat stress. It seems more so than the hard corals are. Another one of my master's students, Jen Magel, was really interested in looking at the fish. So in addition to photographing corals for many years, we've also been doing underwater visual censuses of the fish communities since 2007, counting hundreds of thousands of fish along the way. And Jen wanted to know what the impact of the heat stress was on reef fish. And I thought that was kind of interesting, but also was so focused on the corals at the time that I didn't really foresee what a huge impact it would have had on the fish. So I kind of turned from fish to having a one-track coral mind by this point. Jen brought me back to the reef fish and did some beautiful work that we published in Ecological Applications, showing that in July 2015, 2 months into the heat stress event, the reef fish entire community abundance and biomass had plummeted by about 50%. So reef fish are also living quite close to their upper thermal tolerance limits, and they did not like the heat stress. I mean, that was only heat stress of between 1 and 1.5 degrees Celsius, so not a lot of heat stress one might think, but they had a very strong reaction to it. Our understanding is that they mostly moved deeper on the reef because these populations did bounce back pretty quickly. Okay, so what are some glimmers of hope? So to think about the glimmers of hope, we are going to turn back to all of those tagged corals that I talked about. That was my plan for studying the impacts of this heat wave. And so we did end up tagging many corals around the island, over 1,000 in total, and we tracked as many of those as we could throughout the entire event. And I'm just going to show you a couple of things that we found here. So this is a coral that these are photos taken at the end of the event. So in that March, April, after 10 months of heat stress. So a lot of the corals were bleached at that point, but in subsequent years, we found that they had recovered. There were also definitely oddball corals. So this is a branching coral, Poecilopora. It did not fare well overall as a species. It was highly susceptible, but we also had Some colonies that at the end of the event weren't even bleached, and they flourished after the event. So I fully understand that you have to be a very glass half full kind of person to think that 10% survival is a good news story and provide some glimmers of hope. But I think there is a lot that we can learn scientifically from trying to pinpoint why some of those corals were able to persist that very prolonged heat stress. And the most surprising thing that we found occurred in these corals. This is Platygyra rutensis, known as the brain coral. And it did something completely unexpected that we were only able to pick up on because of having tagged these individual colonies and followed them through the event. So this is a depiction of the heat stress that unfolded on Christmas. The dashed vertical lines are the various expeditions that we did. I don't know if anyone else studies El Niños in the room, but the 2015-2016 El Niño was originally predicted to come a year before. It was meant to be coming in 2014, and we got funding to go out and study it. And so we ended up doing a lot of expeditions that all ended up being before the heat stress came. So the El Nino came a year later than predicted, which means we have a lot of great before data. So this is the timing of all of our expeditions through the event and afterwards. And I'm just going to take you through one representative brain coral colony to show you what this species did. So prior to the event, we were tagging colonies. They looked okay. Early on in the event, they were bleached, which is not unexpected. And then what we predicted, and every coral biologist would have predicted, would be that if these corals were going to survive the event, that they would only be able to reestablish symbiosis after the heat stress had abated. But instead, what we found was that these brain corals reestablished symbiosis while still under heat stress. So this is completely counter to everything that we coral biologists think we know about how symbioses work. Kind of completely turned our understanding of these symbioses on their head. We found this in many different brain corals, as well as patchily in a few other species. Okay, so how did this happen? How were they able to do this? And so to understand this, we need to go back and think a little bit about their symbiotic partners. So this is a recent taxonomic revision of the entire group of Symbiodinaceae. And I'm just going to point out symbionts that are at two ends of the spectrum, really. So Cladocopium, the genus Cladocopium, also known as Clade C. It's a very diverse genus of symbionts. They're often regarded as being the ideal symbiotic partners. I always think of them as being the type that you want to be in a relationship with. They are a little environmentally sensitive, but they also are very generous symbiotic partners, providing their coral hosts with high levels of energy. And at the other end of the spectrum, we have the genus Durastinium, Clade D. These are on the plus side. Again, there's always life history trade-offs. On the plus side, these are heat-tolerant symbionts, so environmentally robust, but they also do not provision their hosts with very much energy. And so there are these very strong life history trade-offs. So what we found was that the dominant symbiont type in the brain corals prior to the heat stress event dictated whether or not they survived. And so if you had to make a prediction about this, you would probably predict that it was those brain corals that started with Clade D, the heat tolerant symbionts, that would have survived. But that is not what we found. We actually found the complete opposite, that survival of brain corals happened when they were initially dominated by those heat sensitive C symbionts. So what you're seeing here, I'm going to show you a progression of these symbioses. This is all for the brain coral. And what the measure here is symbiont to host ratio. So it's the number of symbiont cells to host cells, which is really a measure of if the coral is in a state of healthy symbiosis, they've got this high ratio of symbiont to host cells, versus when they're bleached and they've ejected their symbionts, they have a very low ratio of symbionts to hosts. So the corals that started with clade D symbionts, these heat tolerant symbionts, early on, both types, early on, so just at the very beginning of the heat stress, brain corals that had C or D were in an okay, healthy state. But 2 months into the heat stress, those that were dominated by D still hadn't bleached, which makes sense. Those are heat tolerant symbionts, so they still hadn't bleached. But that's the last time point that we have for those, because after that, they all perished. In contrast, the corals that started with the heat sensitive symbionts, Cs, in July, a couple of months into the heat stress, they had bleached. But by the end of the heat wave, they had reestablished symbiosis with those D symbionts. And in subsequent expeditions, and we sampled those ones, and that new type of symbiosis persisted. So there's one more facet to this story, and that is that it was not random where these brain corals, or what type of symbionts these brain corals had. It was very spatially segregated around the island. And it had to do with the underlying level of local disturbance. So these are the sites around the island where we had tagged colonies of the brain corals. And this principal coordinates analysis is showing you differences in the community composition of symbionts within those brain corals. And what you can see is that the brain corals that were exposed to very high levels of underlying local anthropogenic stress, so the corals that were up here, right where the villages are, they clustered separately from most of the corals that were exposed to lower levels of local disturbance, with a couple of exceptions. And so the corals that were exposed to very high levels of local disturbance all started with D symbionts. And almost without exception, they all perished. The brain corals that were exposed to any slightly lower level of local anthropogenic disturbance almost exclusively started with C symbionts, and most of them persisted throughout the event. So this provides some interesting new evidence that underlying levels of local disturbance actually can influence climate change responses. And although I think conceptually it makes a lot of sense, a lot of us would think that if we stress out reefs a little bit less, they'll probably be able to be more resilient to future stressors. But in fact, this has been an area of really active debate within the coral reef world. And so this is some evidence that local conditions do matter. So of course, during COVID, we have not been able to get to Christmas Island. And so we started gazing in the ocean a little bit closer to home here on the west coast of Canada at our kelp forests. And that was not coincidental. It was, I guess, two reasons. I have a very talented postdoc, Dr. Sam Starko, who is a kelp expert. And I had hired him to work on corals, but he never made it to a coral reef. And so I don't know, I think he's taught me as much about kelp as I've taught him about corals over the past couple of years. But scientifically, another reason for starting to study kelp is that definitely tropical corals are the most vulnerable marine ecosystems to climate change, but kelp forests are also highly vulnerable, and they're the most vulnerable temperate marine ecosystems to climate change. So what I'm going to present to you here is work really led by Sam and his main research collaborator, Chris Neufeld, that's been done out at Banfield Marine Sciences on the Sciences Center on the west coast of Vancouver Island. And this is a study that we've just got in review now. And it's really focusing on microclimate. So microclimate predicting kelp forest extinction through heat waves. So until I started working with Sam, I always used to say that the heat wave that unfolded on Christmas Island was the most intense heat wave ever experienced in the ocean. And Sam always likes to correct me and point out that actually during that very same heat wave, there was more intense and more prolonged heat stress felt right off the coast where I am here. So the kelp forests were actually experiencing an even more intense heat wave. That was partially got known as the blob out here. And it was indeed more intense than what was experienced by corals. So Sam and Chris were really focusing on a gradient, a microclimate gradient that I'm going to show you in a minute. And it relates really nicely to this paper, Biologists Ignore Ocean Weather at Their Peril, by my new colleague, Amanda Bates, where she points out that often in the ocean, we're focusing at really, really large scales, but that there is also very fine scale environmental variation, not just temperature, but many other environmental factors that are happening on very, very small spatial scales, and that it's important to understand biological responses on those scales as well. Okay, so I'm just going to orient you to this map. We're looking at Barkley Sound up here on Vancouver Island. This is where Bamfield is located. And within this area, you've got the open ocean coast here. And then you've got this microclimate gradient up Barkley Sound. And so this is about a 16-kilometer stretch of coastline, so a very short stretch of coastline, but a stretch of coastline that over this very small geographic area, the temperature spans 3 degrees Celsius. So a huge temperature difference across just a very small spatial scale. Chris and Sam have pulled together a number of different data sets, historical data sets on kelp presence, and more recent ones, and then runs throughout and since this heat wave. And what they've shown is that throughout this heat wave, the kelp that were exposed to microclimates that were cooler mostly persisted. So these are all the blue sampling areas, whereas kelps that were exposed to the upper end of this temperature gradient almost all perished. So just to depict this one other way, this is the probability of extirpation. So this is for Macrocystis, the giant kelp, and Myriocystis, the bull kelp, and a separate data set that combined the two of them. And what you can see is that the probability of extirpation of these species increased significantly as you move from the open coast in towards this higher level of the microclimate gradient. So sort of parallel situations going on with very, very vulnerable kelp species and very vulnerable coral species. And of course, these species are biogenic foundations of these ecosystems. So if we lose these biogenic foundations, we're really at risk at losing a lot of the other components of these ecosystems. So I'm just going to finish by showing you a little bit of the new work that we're embarking on now. We had our first full lab field season out at Bamfield Marine Sciences Center this past summer. This is led by Sam and Chris and one of my new PhD students, Matt Sordas, who you see here. We're doing a lot of broad environmental monitoring out at Bamfield. And one of the projects that we're looking at is with this species of kelp, intertidal kelp, canopy forming kelp, Pastelsia. We have long-term data sets for this species, and so we're looking at long-term changes in this species. And I basically just can't resist showing you this. This species of kelp is known as the sea palm, and it has become my favorite species of kelp because it looks like a little palm tree and is absolutely adorable. Okay, so that's it for the impacts side of things. If you're feeling a little bit depressed about all of the species loss that I just told you about, you're certainly not alone. I sat with this for several years, and about a year ago developed a new course at UVic looking at ecological impacts of climate change in the ocean. And I taught it last winter semester. And as I was teaching it and going through the litany of assaults, ecosystem by ecosystem, something in me kind of snapped, I think. I just reached a breaking point of, I can't keep doing this. I've been documenting impacts, human impacts in the ocean for almost 25 years now. And I want to start focusing on solutions and thinking about ways that we can actually improve things. And so one of the ways that we can think about that is actually thinking about the fact that the ocean is not just impacted by climate change, it also presents a great opportunity for climate change solutions. And this was highlighted in a recent report by the high-level panel for a sustainable ocean economy, led by Oth Goldberg, who's a coral reef ecologist as well. And in this report, they point out the many different ways that the ocean can contribute to climate change solutions. One of the biggest ones would be if we ramped up renewables, marine renewables, so wind and offshore wave energy, reforming the shipping industry, reforming our fisheries, reining in fisheries subsidies, for example. So transforming fisheries and aquaculture, but then also thinking about the protection and the restoration of natural climate solutions. So blue carbon ecosystems that are natural carbon sinks. Now, interestingly, this has been getting more and more recognition, and over 75 different countries around the world have now incorporated these types of ocean-based climate change solutions into their portfolio of climate solutions. And yet Canada, which has the world's longest coastline, has not yet incorporated ocean solutions into its portfolio. So my longtime collaborator and friend, Susanna Fuller, and I, who's many of you probably know and is based on the East Coast, works for Oceans North. We wrote an op-ed about this for World Oceans Day this past June, highlighting how if Canada actually harnessed its oceans, it could really help getting to our climate change goals. But moving beyond that, since I guess I'm finally at an age where I want to actually start working on solutions, not just be cranky at DFO, which is a pretty easy thing to do. So we're starting to actually work on doing the full accounting for the entire country of the potential for natural climate solutions in Canada's oceans. And so in this project, we're really asking to what extent Canada's oceans can provide natural climate solutions by calculating the extent of all of our different blue carbon ecosystems, the amount of carbon that they store and sequester, and then also working with climate scientists to project how those ecosystems are going to change in the future under different climate change scenarios. And so this is a broad collaboration with Susanna, with a number of different scientists and managers at Fisheries and Oceans Canada and Parks Canada, with BC Parks and a number of different NGOs, including Ecology Action Centre in Halifax, which I know many of you are familiar with. And so we're just embarking on this new work. We will be starting it in the new year, really getting in full swing of it by the spring. And so if any of you are getting close to finishing your PhDs or looking for postdocs, we'll be hiring several postdocs for this project in the spring, so you can keep an eye open for that. I'll just finish this article. Actually, just this is very good timing. It just was sent this morning, so it just actually came out this morning. A new article about the importance of oceans for Canada and for Canada thinking about climate change solutions. So that's in the National Observer today. And I will finish there with a quote that sometimes keeps me going from Christiana Figueres. Optimism means envisioning our desired future and then actively pulling it closer. Thank you.